about it without, say, if you, what's a good example? So if it was no longer identical, say there is a couple chromosomes happened over here and a couple didn't happen over here, the one that's missing, or maybe has too many, might not be able to function normally anymore and the cell dies. So a really important part is to make sure in the process that it's separating equally. And so it goes back to 1N instead of 2N. Now I kind of made a jumble of that. Do you guys understand that at all? Do you get the concept that when you replicate it, you have 2 sets, so it's 2N, and then when it's unduplicated, when it goes into the 2 separate cells or 2 daughter cells, that it's 1N. So it goes back to having the normal amount of chromosomes. Is that clear? Yeah?
Yeah.
Perfect. Okay. So, and then also as well in this diagram, it's kind of nice. You can actually start seeing the spindle fibers disintegrating. They're going away. Everything's reforming. So soon, pardon me, soon you'll be able to have the 2 cells. And like I said, cytokinesis is happening simultaneously. So as telophase is happening, cytokinesis is separating the cells, and now you're going to have 2 individual daughter cells from the original parent cell. But yes, and then so that's kind of the basics of mitosis. And here, sometimes it's hard to actually visualize what happens. And this is kind of a nice little video that I found that really shows, you can see it lining up and being pulled apart, and you can see the lines starting to form for the cytokinesis to happen. Okay, so can you guys, does that help with the visual? So what's happening right now? Do you guys know? So at the beginning, everything's lining up, being pulled apart, and then you can see the cleavage forming. So now that we kind of have a basic understanding, I want you to address some of these questions. So what types of cells go through mitosis? As well as, do both plant and animal cells go through mitosis? Why is the cell cycle necessary? Why do we even need it? As well, I want you guys to research further into detail. I want something that from this lesson you might not have found totally clear. Maybe research maybe the concept of 2N versus 1N. Maybe it wasn't explained well enough. So do a little bit of research. It should only take about 5 minutes. Find a question, and then next class, I want us to address and answer these questions. And as another extra little bit, I want you guys to be able to identify which phase things are going through. Please feel free to look online to help you guys figure it out. And I want this to be the beginning of next class. We're going to go over our questions. We're going to go over which answer is for which cell. And we're going to lead into meiosis. Do you guys have any questions?
No.